Okay, students. So I discussed about few of the experiments associated with the DNA, actually the transforming phenomena, then the chemical nature of the particular transforming factor. In the same context, I have discussed today related to the Hershey and Chase experiment. So I told you that people used to carry out the experiments which was conducted by the earlier scientist. So the work of Avery, MacLeod and McCarthy was further taken up by Hershey and Chase. What happened is that in 1952, the Alfred Hershey and Martha Chase, they have worked onto the Avery, MacLeod and McCarthy's evidence, and they proved that DNA was actually the causative genetic material that transfers from one generation to next generation. They have done a blending experiment where they have used the bacteriophage named as T2. It is sometimes also called as phage. So they have used the radioactive isotopes such as phosphorus 32 and 35 sulfur. Right. So I have told you about that this is the mechanism behind the attack of the bacteriophage. The bacteriophage once attaches onto the cell. This is the basic phenomena of attachment of this bacteriophage onto the cell. You can see they make a pore here at the cell surface, and from this pore they send their genetic material inside. The red colored thread inside appearing is a genetic material. This is a generalized viral constituents. So a genetic material and a outer capsid coating. Now once the cell phage attaches itself, it sends its genetic material inside, and this genetic material is replicating constantly, making its further progeny, and it goes out. So I mean, from one to making a number of copies. This is also called as lytic cycle of bacteriophage. So this is a general life cycle of a bacteriophage. So then the experiment which they carried out was that in both of the cases, first you see this first set of experiment where the bacteriophage used to infect the bacteria in a which was growing in a medium which contains 32 phosphorus isotope, and this phage was when who this phage which infect the E. coli actually is replicating, and then means its genetic material gets replicated. So 32 phosphorus gets incorporated into the genetic material, that is DNA is having maybe DNA or RNA, whatever it is. So it is having a phosphate. So the phosphate enters over there. I mean, the entry of the phosphate isotope makes is possible very much over there. So they carries on this genetic material, and this genetic material keeps on replicating and moving on from one generation to the next generation. So what happened ultimately is that when they after performing the experiment for one or two generations, they do the blending experiment to remove all the outer coatings of the bacteriophage, and find out that the radioactivity was very much available in the host, and this was able to pass on from one generation to the next generation. So this is a proof that DNA is the genetic material, because 32 labeled phosphorus has added in the media. Phage infects the bacteria, so phage uptakes that 32 phosphorus in its genetic material, and when phage attaches to the next bacteria, it transfers that radio labeled genetic material which keeps on multiplying and reaches to the next generation. Similarly, they also tried the experiment with 35 sulfur. So they infect the E. coli in a sulfur containing medium, and they obtained that in that sulfur containing medium, when they phage after infecting, since sulfur is actually a main component of amino acids, and amino acids are building blocks of protein. So you know that phage is having an outer layer of protein. So that protein is now coated with that the presence of 35 sulfur was observed in that outer left out protein onto the cell. So because of that, it was confirmed that protein could not be the genetic material, as it remains outside of the cell. Okay. So now, as per the timeline which I have shared earlier, Mixture, we know it associated with discovery of nuclein, then Griffith, then MacLeod, McCarthy, then Hershey and Chase. So all these experiments are associated together. Now in my next uploading slides, I will be describing the work of further scientists. Thank you.